Before we jump into the topic, let's explain what it is. DNA replication is the process by which DNA makes a copy of itself during cell division. Depending on the type of cell, it can occur in different sites. For example, in eukaryotes, DNA replication takes place in the nucleus, while in prokaryotes, it's in the cell cytoplasm. To start off, DNA replication starts at the specific location called the origin. This site is usually identified by certain DNA sequences. There, we are introduced to our first enzyme, helicase. Helicase would help unzip the DNA strand into two separate strands, and they can do it with ease by breaking the hydrogen bonds that hold the DNA bases together. In order to keep these strands separated, SSB proteins, or single-stranded binding proteins, will bind to the strands to keep them in place. After, our next enzyme, topoisomerase, comes into play. Topoisomerase will be the enzyme that will prevent supercoiling, which is important when it comes to DNA replication. If not prevented, it can cause an overwinding of the DNA, and the next step would not be possible. Once split, the two strands will be categorized in two different parts, the leading strand and the lagging strand. How can we determine which is which? We'll get to that in a bit. Once separated, the primase, which are enzymes that help make primers, will be present to help guide DNA polymerase to determine where to start replicating. DNA polymerase is an enzyme that helps replicate DNA molecules using the old DNA strand as a template to help build a new strand of DNA. DNA polymerase will replicate DNA molecules from a 5' to 3' end, which can be seen in this picture. This means that it will add new bases on the 3' end on the new strand. As DNA continues to unwind and builds these bases, DNA polymerase functions differently on specific strands. This is where we're going to talk about the leading and the lagging strand. Since DNA polymerase can build new strands from a 5' to 3' direction, it becomes difficult for one strand and the other as the position of a 3' and 5' are opposite in both strands. Therefore, due to this motion, there will be one strand in which replication is continuous and the other strand being replicated in fragments called Okazaki fragments. The strand in which replication is continuous would be classified as the leading strand, while the strand that is replicated in fragments is classified as the lagging strand. Once replicated, the ligase will come in to help the lagging strand. The ligase is the enzyme that helps put DNA fragments together in the lagging strand. The ligase will fill up the gaps in between those fragments and seal them together as a result. After that, we finally have two new DNA strands built. Something to eye out for is that both strands have one original strand and a new strand. We call this semi-conservative, since the two copies contain one original and one strand. Before it becomes a final product, however, it must go through proofreading, in which errors can be corrected. That is why DNA replication rarely makes a mistake during the process.